Welcome to Study Loop.
To answer this question, we have to have an understanding of the central dogma. So when we see central dogma, we know that this is referring to the transfer of information within biological systems from DNA to RNA to protein. And this is a very essential concept that is key to understanding molecular biology and biology on a molecular level, specifically referring to genetics and how genetic information is going to be transformed, really, from one form, like DNA, into another, RNA, and then into another form, protein. And of course, we know that from DNA to RNA, this is going to be transcription. And from RNA to protein, that is translation. So now we have to understand what kinds of viruses will adhere to this flow of information and what kind of viruses will not. And we also have to understand that there's a few different types of viruses. There are DNA viruses, there are RNA viruses, and there are retroviruses. And retroviruses are going to be RNA viruses as well. So to answer this question, let's just go through the different types of viruses and understand what kind of form their genetic information will be stored in. So in DNA viruses, the form of information storage for the genetic information is in DNA. For RNA viruses, it's in RNA. For retroviruses, it's also going to be stored as RNA. However, for retroviruses, what's going to happen is that the RNA is going to be transferred or transformed into DNA. And this is done through the process of reverse transcription. And as you can see, if you look up here, DNA to RNA, we said that this was transcription. Well, if we go in reverse, then this is exactly what we have down here, reverse transcription. Now, for our DNA viruses, we know that the information is stored as DNA. That means that it must then be transcribed into RNA, and then it must be translated into protein so that we can produce viral proteins and viral RNA. And this is all going to be important for viral replication. For RNA viruses, we are going to harness the cell's translation machinery to produce viral protein. And we will also use our RNA strand that is part of the virus's original genome. So we know that the genome is going to be made up of RNA. So it will use its genome and it will replicate its genome by using one strand of its double-stranded RNA as a template, and it will copy it over, and it will just produce copies of that RNA genome. So it will also be able to replicate its RNA genome through a mechanism that is quite similar to DNA replication, which we find in just any eukaryotic or prokaryotic cell that will be undergoing mitotic division. So it must replicate its DNA first. So the same concept will apply to RNA replication. So it will also be able to produce viral RNA. The final thing that we have to understand here is what each kind of form of the virus, the DNA virus, the RNA virus, and the retrovirus, what are some examples of these viruses? So for our RNA virus, we are looking at influenza. Okay, so influenza is an example of an RNA virus. Whereas for our retrovirus, that would be HIV. So therefore, the correct answer choice here, putting all this information together, is answer choice A.